Let us get started. We'll start by going over the warm-up. And again, remember, the next time we meet, it's going to be your first major exam on Tuesday. It's going to include everything from first day, all the chemistry, up to all the stuff we've been talking about with action potentials and graded potentials and all that. So here we go. I'm gonna share this. Okay. So the first one, all that are true about action potentials. Whoop, that's not what I meant to do there. Can travel long distances. Remember, these are the ones that can go like, a meter going from like your spinal cord all the way to the tip of your toe. So definitely positive or negative. That is not, we talked about an action potential is kind of like that wave. So it's like an all or none kind of thing. There's no such thing as a positive action potential or a negative action potential. It's either going or it's not. Kind of like a fuse is either lit or it's not. Occurs whenever a receptor binds a neurotransmitter? Most definitely not. We saw there's usually the influence of many synapses all working together and all kind of adding up to bring the axon hillock above threshold. Occurs mainly on axons, most definitely. Tends to be localized to a specific spot, most definitely not. They travel long distances. That chain reaction goes along the whole length of the neuron. Mainly on the cell body, nope. Mainly on the axon. And created by special voltage-gated ion channels. Obviously, you spent a lot of time talking about the voltage-gated sodium channels that open fast and then inactivate, and the voltage-gated potassium channels that open slowly and close slowly, and how together those create that classic looking action potential spike. Graded potentials, on the other hand, do not travel long distances. They're just going to be local part of the membrane. Positive or negative? Yes, you can. Again, we talked about excitatory postsynaptic potentials, inhibitory postsynaptic potentials, depending on what kind of ions are crossing the membrane in which direction. The voltage might go up, it might go down, might go up a lot or a little, or down a lot or a little. That's continuously variable is another adjective we use a lot for graded potentials. All or nothing, no, that's action potentials. Tend to be localized, that is true. Are created by specialized voltage. These are not made by voltage-gated channels. These are more about ligand-gated channels. These are when a neurotransmitter is released into a synapse and binds a receptor and opens up channels. So this would not be voltage-gated channels. Occurs mainly in the dendrites as well as on the cell body. Yep, not on the axons. So if you are still kind of, definitely before Tuesday, you want to be like crystal clear about what's the difference between graded potentials and action potentials and where you find them and what their roles are. When a neurotransmitter is released into the synaptic cleft and binds with the receptors on the postsynaptic neuron, here most people got, it might excite or inhibit, like I was mentioning, depending on what kind of ions are being allowed to cross the membrane. It might make the voltage a little higher, bring everything closer to threshold, or maybe make it more negative, take it further away from threshold. So it does not, definitely doesn't damage the neuron. Because that's the typical way that neurons are just kind of talking to each other. You'd be in trouble if it caused damage. Describe at least two different ways synapses have cleared of neurotransmitter. Most people got this. Again, Okay, there's three main ways we talked about. What are three main ways to clear a synapse of neurotransmitter?
Enzyme.
So one is using enzymes to break them down.
Reuptake.
So reuptake, kind of getting vacuumed back up into the presynaptic terminal to get reused or recycled. And then just kind of diffusion out of the synapse. So, and again, it's important that we clear the synapse so the message turns off. This one, the large depolarization upward swing in membrane voltage during an action potential is due to, and most people got this, we talked about, remember when those fast voltage gated sodium channels open and we have sodium rushes in, and that is bringing the voltage on the membrane close to that equilibrium potential for sodium. So it's sodium rushing in to create that upward part of the spike, the volt of the action potential. So this was the classic action potential. Kind of goes up and resolves. We often do call them spikes because it looks kind of like a spike of voltage going up. Like when I used to stick electrodes in frog nerves and measure the activity, we would talk about them spiking. If you're actually just looking kind of more at a what's going on with the neuron, it just looks like wherever a action potential is running down, it looks like a big spike of voltage going up. So we often talk about spikes, and you can actually hear it. We put basically put a little amplifier with a little speaker on it, and you could just hear this kind of pop whenever the axon fired off. So it was, you could actually, I was particularly looking at axons from the inner ear that were coming from sensors that detected vibration. So you could just kind of vibrate the frog a little bit, and you could just hear this pop, 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 as the action potentials would start running down the axon. So, so this is going up as NA is rushing in. What is causing the resolution, the coming back down to the resting?
Potassium entering.
Yeah, so there's two things. One is potassium is entering, which is bringing us back down to that negative. And then what else happened to kind of stop the influence of the sodium?
Sodium gates close.
Yeah, the sodium gates. And if we want to be kind of more precise, we would say the sodium gates inactivate. Since we're still, remember threshold, we should put our threshold in here. As long as you're up above threshold, the sodium gates wouldn't officially be closed. They're like closed when you're down below threshold. But what happens is the sodium gates, they open fast and then inactivate after a millisecond. Right, so they open and allow the voltage to swing up, but then they inactivate. And until they have a chance to totally reset, they are now gonna not have any conductance. And again, as somebody was saying, as soon as these guys are inactivating, we have the potassium rushing in, and that's bringing the voltage back down. And in fact, we have a bit of an undershoot because we have a higher amount of potassium conductance than normal, which makes the membrane voltage even more negative than normal resting. And eventually we kind of get back to resting. So any questions about just the basic mechanics of how an action potential happens? Cool. And again, remember we've got our overall neuron. We have different things talking to this one. Bring in the voltage up and down here at the hillock. And as soon as we fire off an action potential here, that positive depolarization there is going to excite the next little piece of membrane, which is going to excite the next little piece of membrane, which is going to excite the next little piece of membrane. So you just got this wave traveling down to the boutons, the end, the axon terminals. A few other things we can talk about here. When we're looking at the graded potentials that are influencing the overall voltage on this neuron, and ultimately, remember the axon hillock is where we actually really are integrating it all. Axon hillock, the sum of all the IPSPs and the EPSPs. So there are two ways that these dendrite or these axons, these other neurons coming in to talk to this neuron, can actually kind of work together to have more of an effect than they might on their own. So let's talk about spatial summation and temporal summation. So far, I've just kind of made this picture here where this is going, or this is going, or this is going, or this is going. And we can talk about, let's say we have, I'll put a black one and a green one. If these are close to each other, and they are being fired off, we're going to have a larger overall effect than one or the other would have had on its own. Right, so let me maybe draw another picture here. So let's, so here's my black one coming in, here's my green one coming in. This is just my overall neuron. And again, these are close together. So I'm going to talk about spatial summation first. And I'm going to look at V sub M, just the membrane voltage on our neuron here. And again, it's, let's say this is minus 70 millivolts. It's kind of going up and down or whatever. If we fire off, let's fire off this synapse here. There's going to be, let's say it's a excitatory postsynaptic potential. It's going to open up some sodium channels. Then what's going to happen, we fire this off, the voltage is going to go up because we opened up some channels. Number two, I could fire off, and it's also going to cause some depolarization. The membrane voltage, maybe I should get better with my coloring here. Here, I'm going to make the neuron they're talking to red. I'm realizing my color coding is kind of wonky. So red is my main neuron. Here I'm firing off this little black neuron that's talking to it, and it has some effect on the overall membrane of the neuron. I fire off my little green neuron here. It has some effect. If I fire off 1 and 2 together, and they are also located in the same space, you can imagine what's going to happen. Each one is letting in, cations, and together they're going to let in even twice as many cations as they would have either one on their own. So the overall effect is a larger depolarization of the target membrane. So this is what we call spatial summation. It's when we have two things that are located, they're on their target on the membrane is really close, and then they're working together, they can have a larger overall effect. If you want to think about this in terms of just practical like circuitry, this is like a coincidence detector, right? If you're trying to decide whether different things are happening at the same time, right? If just 2 is going, you're not going up very much. If just 1 is going, it's not going up very much. But if 1 and 2 are happening at the same time, then we have information that there's this coincidence that these things are happening. There's some kind of synchrony going on, right? So this is a way, like kind of a way in the neural circuitry to detect that there's activity from both of these at the same time, because the overall effect is going to be a larger depolarization, more likely to trigger an action potential in the target neuron. Does that make sense? And in the idea of synchrony, I'll just mention this word. Synchrony meaning happening at the same time. It's pretty important, or it's pretty clear that that's important in kind of the functioning of your nervous system, particularly your brain. And we'll talk about kind of big picture things, but one of the most interesting things people look at is like consciousness. What does it mean to be conscious? And that's something we're still really at the very beginnings of trying to understand. But what does seem to be clear is synchrony, having different parts of the brain kind of working at the same time is part of the information that kind of binds. We talk about the binding problem. By binding, I mean, how do we connect all these different parts of your experience into the you that is experiencing this right now, right? There's sound information being processed by your auditory cortex and visual information by your visual cortex, and all your memories that are part of your autobiographical sense of who you are. And somehow that's all getting bound together into this moment of you listening to me, watching your screen, having this moment, and it's not clear still exactly how that happens or what it means to have that experience, but it seems pretty clear that this idea of synchrony plays a role, having things kind of spiking together and being kind of, not just kind of random things, but kind of happening in kind of these synchronized waves creates some part of the answer to the puzzle of how are these things all getting tied into the same experience. So blah, blah, blah. Spatial summation. We will see this again a little later, particularly when we start talking about one of our labs later. There's another kind of summation I mentioned, temporal summation. So let me put that in here. So again, I'm going to kind of draw my picture, some neuron, some hillock. And again, there's some other neuron that's talking to it. But at this point, for temporal summation, we only have to think about one other neuron that's talking. What does temporal mean? If something has to do with temporal, what does that adjective refer to?
Time.
Time. Yeah, temporal means having to do with time. Right, that's why these are your temples, like your temples or your temporal lobe is because this is where your hair starts turning gray, indicating the passage of time. If you watch Star Trek, you've probably seen lots of temporal distortions or something where, whoa, time isn't running the way it should be normally running. It's slowing down or whatever. Anyway, so temporal summation. If we look at just this yellow neuron fires off, kind of like we've been saying, there's going to be some change in my cell membrane here as I puff my neurotransmitter on there, open some channels. And if I hit it again, I'll get another little blip. Temporal summation is what if I hit this one in quick succession? So boom, but then boom. So the same synapse, but I'm hitting it quick succession. So I'm letting in some cations, but before it has a chance to resolve, I hit it again and it kind of dog piles. So temporal summation is this kind of dogpiling of the stimulus. If you fire off this synapse in quick succession, the overall effect is going to be a larger depolarization at the target membrane than they would have been just from a single triggering of that synapse. Does that make sense? Yeah, keep this in mind. When we do our nerve stimulation lab, this is going to be at the core of kind of developing our hypotheses about what we're going to see. This idea that if you hit the same membrane, but in quick succession, you're going to basically be adding up or having more of an effect than you would have just from a single stimulation at that location. So temporal summation, what do they say? Put it on the shelf or whatever. It's going to be coming back. You don't want to forget it. I should mention, okay, let's say at this point, so stopping point for exam 1. So just want to make this kind of official thing of like, I'm going to continue on. We're going to start digging into the nervous system proper right now, but that's on exam 2. So everything up to this point could show up on exam 1 on Tuesday. But everything we're about to talk about is going to be on exam 2. So everything from here to the end of lecture is on exam 2. So just kind of putting that out there so people, there's already plenty of stuff for you to study and be responsible for. The last thing you need to do is kind of spend time trying to remember the details of other stuff we're talking about today that you're not actually responsible for. So, all right. Let's do this then. So what's kind of exciting is we are going to finally introduce our first official system. We've spent a month already in this class and we haven't actually talked about any organ systems. So we are going to start with the nervous system. And we'll start by kind of laying out, like, what is it doing? What is the overall kind of geography of this? And then we'll kind of be spending the next month looking at details of different parts of it. So what is it doing? What are functions of the nervous system?
To receive stimuli from the rest of the body.
Yeah, that's a huge part, sensory. So we call this the sensory division. We also call it the afferent division. So, in the sensory or afferent is stuff you're more familiar with, like the five senses, like vision and touch and taste. But there's also all sorts of senses that are more internal, like what's your blood sugar doing? What's your temperature? How stretched out is your stomach and your intestines in the moment? What is your blood pressure? Right? So there's the sensory, this is both kind of external and internal. Just kind of making that clear. You have the senses that are, you think of more kind of classic five senses, but a lot of sensory information is coming in that you are not as aware of moment to moment. Like you're not thinking about your blood pressure, but your brain sure is tracking it and keeping it in control, right? So you are not aware. We're going to talk about the difference between perception and or what perception is as we go on. Perception is kind of like a conscious awareness of stimuli. And that's a whole other kettle of fish. So sensory or afferent. What else does the nervous system do?
Send instructions.
So send instructions. So we activate things. We're going to have what's called the motor division. So this is also called efferent. And this is going to activate, basically it activates muscles, activates glands. And again, some of the muscles are muscles that you think of like, oh, I'm going to kick the ball. But other muscles are muscles you're not thinking about, like changing the diameter of your blood vessels to control blood pressure, or squishing little things to squeeze stuff out of a gland, or maybe changing the focus on the lens of your eye or the diameter of your pupil. So there's going to be muscles that are like the voluntary, the skeletal muscles, as well as the involuntary, the smooth muscles. And again, glands, just turning on a sweat gland or an endocrine gland or who knows what. So that's going to be part. And then the part in the middle, we call integration. Integration means kind of figure out, what do? We have all this information coming in about the world around us and the world inside of us. And typically you have to decide what is the appropriate response, right? My blood pressure is going up. I need to activate some stuff that's going to bring it down. Or, somebody is waving at me, I gotta wave back at them. There's who knows what, right? There's going to be some information coming in, and then you the integration is kind of figuring out what is the appropriate response, and then there is kind of this response. We're going to look at what I've drawn here is actually a classic reflex arc, but it's at the core of a lot of your nervous system functioning is this kind of reflex arc where there's some information coming in, you process that information, and then have some kind of appropriate response. Again, well, in fact, our lab on Tuesday is going to be the reflex lab, which is going to just be looking at this in more detail, this idea of reflex arcs. The integration, the kind of figuring out what to do with the information that we're getting is typically the brain and the spinal cord. It's not always. As we continue on, we'll see that the digestive system has its own integration just within the walls of the GI tract, the whole enteric nervous system. But for a good first approximation, you can think of the brain and the spinal cord as being the main things that are figuring out how to kind of run the ship, so to speak. And we say the brain and spinal cord collectively, we call the CNS, the central nervous system. You know, that's where we're going to actually be spending the next couple of weeks talking about, is just looking at the brain and the spinal cord and what they're doing and what they look like, blah, blah, blah. The sensory and the motor, collectively, we call the PNS, or peripheral nervous system. So PNS, peripheral nervous system. The mnemonic, in case you haven't seen this before, the mnemonic for remembering afferent versus efferent is you think about efferent with the E, the stuff that's exiting from the CNS. The afferent with an A, that's the stuff that's arriving at the CNS. So that's like a nice little mnemonic to remember afferent, because I will use those interchangeably. Efferent nerves, afferent, you need to know efferent are motor, afferent are sensory. So that is terminology you do need to know. So this is the biggest picture here. Again, didn't mean to do that. You've got the CNS, you've got the PNS, there's a bit more divisions that we can make, particularly with the motor side. So we're going to do, as we're introducing the nervous system, so you have a kind of complete roadmap of the different sections that we'll be looking at, I'm going to dig a little deeper right now into subdivisions of the motor. So let's do this. So again, we're looking at the nervous system overall. And then we're going to say it's got two main parts, the CNS and the PNS. So that's what we've just laid out for you. Brain and spinal cord. Doing the integration And spinal cord, the spinal cord, even though it does do a lot of just transmitting messages up and down, the spinal cord actually does integration as well. When we look at some of the spinal reflexes next week, things like when you whack on someone's knee and they kick out, you don't need a brain for that. That is being integrated just in the spinal cord. So the spinal cord is actually responsible for integrating some important reflexes that don't need the brain at all. So the brain gets, is obviously got a lot more going on, but the spinal cord is more than just a cable. So PNS. PNS, we said, has two branches. What are the two main branches of the PNS?
Motor and sensory.
Exactly. So we have sensory and motor. And again, the sensory, I said, is both going to be external senses and internal senses. So we'll be looking at this in more detail as we go on. We'll look at all the different senses. How does vision work? How does hearing work? How does taste and smell work? But we'll also be talking about internal receptors, stretch receptors in your blood vessels, in your digestive tract. We'll look at chemoreceptors inside your body, monitoring like your pH or your oxygen and carbon dioxide levels and things like that. So sensory of both the internal state as well as the world around you. The motor is going to be further subdivided. So motor, we have what's called somatic and autonomic. Autonomic. What? Hold on, let me not. Autonomic. All right. Okay, somatic. Soma means body. This is your voluntary motion. So this is motor. This is going to be voluntary, mainly like skeletal muscle. So when we're talking about the parts of the nervous system controlling your voluntary skeletal muscles, it's going to be the somatic motor division. Autonomic, it means self-governing. This is the stuff that you don't think about controlling. This is going to be the involuntary. Like smooth muscle and glands. Right? If somebody says like, like do that cool trick where you make your pupils get big and small and big and small and big and small and big and small. It's like, I can't do that. That's not something I have voluntary control over. So these are muscles that are operated by your, your brain, but, more on these autonomic reflexes. This autonomic has two subdivisions that you need to know about. Actually, wanted to do a different color here. So autonomic is further subdivided into the sympathetic and parasympathetic. All right, let me try that again. All right, barely got it there. So there's two subdivisions here, the sympathetic and parasympathetic. We're going to be looking at all these in much more detail, but in general, sympathetic is what we think of as the fight or flight division. In general, sympathetic is the thing that triggers things that you think of as, getting ready for action, speeding up your heart, dilating your bronchial passages so you can suck down more air, sending blood to your muscles and inhibiting digestive processes, because we don't got to digest our lunch. What we got to do is sprint away from this tiger right now. Dilates your pupils, makes your eyes all wide with fright. So sympathetic, that fight or flight, the adrenaline rush, right? When adrenaline is going to trigger most of these sympathetic actions. We'll talk about what adrenaline is British. Epinephrine is what we're going to call it, epi or epinephrine, but it's basically going to be like a wholesale activation of sympathetic activity in your body. So when you think about sympathetic, think about kind of that adrenaline rush or whatever. Parasympathetic, people often call the rest and digest. It, just from its name, it stimulates digestive processes. It slows your heart down. And in general, these tend to be pretty antagonistic. Like parasympathetic will constrict your pupils, whereas sympathetic dilates your pupils. Parasympathetic constricts your bronchial passages, whereas sympathetic dilates your bronchial passages. Parasympathetic slows down your heart, and sympathetic speeds up your heart. So there's a lot of thing where there's this push and pull, and it's not like your body is in one or the other all the time, right? It's like you're not full on trying to get away from a tiger or lying there watching TV and not doing anything. It's you start moving around, your heart needs to speed up. You got sympathetic activity that is responsible for speeding up your heart. And then you're not working as hard. Parasympathetic will take over and slow it down. Or, so people talk about driving with one foot on the gas and one foot on the brake, and, just kind of differentially letting up or pushing down a little more one or the other. So don't think of these as you're in one mode or you're in the other mode. They're both constantly pushing and pulling and keeping things in some kind of dynamic balance, depending on what your body's needs are at the moment. There's going to be a few things where they don't antagonize each other, and we'll talk about that when we do the autonomic nervous system in more detail in a few weeks. There are some things that are purely innervated by sympathetic. There's a few places, particularly around sexual response, where they actually work together towards a common goal. So, the real story gets a little more complex. But again, for a first approximation, you can think about them as being in this kind of dynamic tension with each other. Over the next few weeks, we're going to be looking at all of these parts in more detail. Again, like I said, we're going to stay in the CNS for the next couple of weeks, probably. Then we're going to be getting into the PNS, and we're going to then be focusing on the motor and somatic versus autonomic, and in the autonomic, the sympathetic and the parasympathetic. We're going to get getting into the sensory and a lot of the details and how your eyes and your nose and your ears work and all that. It's important as we're doing that to kind of keep this big picture in mind so you kind of know how it all fits together. Right? As we go and shine our spotlight on one or the other, remember still there's the CNS and the PNS is the sensory and the motor is the somatic and the autonomic is sympathetic and parasympathetic. That way, if we're talking about sympathetic, you know that we're talking about motor and autonomic motor, and it has nothing to do with sensory, right? When we're talking about sympathetic, we're purely talking about activating things. You know, you might be getting sensory information that is triggering some sympathetic activity that's being integrated by the brain or whatever. But, you know, keeping this big map, this is kind of like our roadmap to the nervous system, is important. So I would, yeah, make sure you like look at this and kind of keep in mind how things fit together. So as we focus in on different parts, you still have that bigger picture of how they all kind of connect into the bigger picture of the nervous system. And then as we continue on, you're going to find the nervous system is inextricably entwined with the endocrine system. Sometimes people talk about the neuroendocrine system, you know, because there's going to be a lot of interplay between the nervous system and the endocrine system. In fact, the brain is directly controlling the hypothalamus, or the hypothalamus of the brain is directly controlling the pituitary gland, which is directly controlling most of your other glands. So there is this real direct and intimate association between the nervous system and the endocrine system as well. As well as, you know, sympathetic. If you want to just have global activation of the sympathetic nervous system instead of activating things through neurons, just dump epinephrine, adrenaline into the bloodstream, and just it'll happen all over the place, right? So just kind of putting that out there, a little kind of teaser. We're going to be having a lot of interplay between the nervous system and the endocrine system. Okay. So we are going to start with just some kind of terminology. So
9 a.m.
Okay. Terms. And I should mention, so again, like I said, we're not in an anatomy class, but you got to have some of the basic anatomy in order to talk about this stuff. So I'm going to just give you some basic terminology that we use to describe things in the CNS versus the PNS. And then we'll get more detail into the actual kind of tissue that makes up the brain and the spinal cord and the nerves and all that. So very commonly, we're going to have this thing where what is it in the CNS and what is it in the PNS? So if I have so if I have axons running along, right, this is going to be the main way you send messages just from one place to another, right? We talked about action potentials going down axons being this way to send messages far. If you have a bunch of axons sending messages in the CNS, in the central nervous system, in the brain and spinal cord, we call those tracts, axonal tracts. And we're going to see different kinds of tracts. We're going to see commissures and projection tracts and association tracts. We'll see different kinds. But basically, if you see that word tract, it's axons running, connecting things in the CNS. In the PNS, we call it nerves. So when you have a nerve, like your ulnar nerve or your brachial, there's no brachial, your phrenic nerve, or your femoral nerve, or your whatever nerve, your median nerve, or your optic nerve. It's just a bunch of axons acting like a cable, sending information from one place to another. That's what that word means in the context of the PNS. When we talk about the sciatic nerve going down the back of your leg, it's just a big fat cable of axons, myelinated axons, sending messages down to the muscles on the back of your leg and sending sensory information back up into your CNS. So that's that. Collections of cell bodies, or I should say neuron cell bodies. So when you have a bunch of neuron cell bodies kind of clustered together in a little island, if you're in the CNS, we call it a nucleus. We're going to be talking about the lentiform nucleus and the caudate nucleus and the amygdaloid nucleus. So it's like bits of gray matter that are located in the CNS, in the brain, particularly when we're looking at it. In the PNS, there are also clusters of neuron cell bodies, and they call them ganglia. So ganglion is one, ganglia is plural. Nucleus, nuclei are plural. So we'll talk about, like, the dorsal root ganglion is the place where you find the cell bodies for a lot of the sensory neurons that are taking messages from your senses into your CNS. There are other collateral ganglia. There's different, when you hear the word ganglion, it means cell bodies outside of the CNS. So these are terms that will come up, so you should probably know what they mean. Let's look at nervous system tissue. It's worth spending a little bit of time talking about how nervous tissue is put together. So far, we have met the neurons. Right? These are the ones that we've been talking about. They're excitable, they're large. They are kind of the rock stars of the nervous system, but it's like any rock star can only do as well as their whole crew supporting them. There are lots of cells that are working in the background to make sure the neurons can do their job. And plus the other ones that do actually really important jobs as well. So there's neurons, but there are also what are called the glial cells. So these are the basically supporting cells of the nervous system. These are the ones that are doing things that are necessary to kind of keep the environment stable. These are the ones making the myelin for the myelin sheath for our myelinated axons. These are the ones that are going to be important for circulating CSF and things like that. We're going to talk about a bunch of different glial cells. Glia comes from the word for glue. Sometimes they're called neuroglia or supporting cells. It's worth introducing some of the main glial cells right now. So, because we are going to be seeing them as we continue on. So let's introduce the glial cells. And like I said, we're going to have often different things when looking at the CNS versus the PNS. So we're going to look at glial cells. We're going to look in the PNS versus the CNS. And there'll be different populations. Both CNS and PNS are going to need glial cells that myelinate axons. Remember, and again, what was the function of myelin?
To speed
Speed up the conduction.
Insulation.
It's an insulation that kind of speeds up the conduction of the action potential. So in the CNS, you have these things called oligodendrocytes. In the PNS, you have these things called Schwann cells. The oligodendrocytes, I'll just kind of leave it there. They make myelin sheaths. You can actually have one oligodendrocyte reach out and make multiple myelin sheaths. The details don't matter so much. You should just know that we have myelinated axons. Schwann cells, I do want to spend a little more time about because they do a little more than just make the myelin sheaths. So if I have an axon, this is my neuron, Schwann cells basically wrap around kind of like a jelly roll or whatever. They leave those nodes there, but each of these is an entire cell. So basically imagine, imagine a jellyfish kind of came up to this and just started kind of spiraling around and just enveloping it in layer after layer after layer of membrane. That's what the myelin sheath is. It's just the specialized membrane of the Schwann cell. Just, in fact, if we do a cross section of this thing, it's going to look kind of like this. You can actually, and then ultimately there's be a nucleus because the Schwann cell actually has a nucleus. So this is one of the main ways. This is the way you make myelin sheaths. Things that are important to remember here is this also plays another role. Imagine you have damage. Remember, PNS, this outside the brain and spinal cord, you cut yourself or you whack yourself really bad. It's not that uncommon to injure. So here is my neuron. Here's the myelin sheath. Maybe this axon is part of your ulnar nerve or something on your arm, and you just got a bad, like some blunt force trauma or whatever, and it's like whack. Oh my God, I just severed this thing. So what happens is, the neuron here is still alive. The axon up to that point is still connected to its metabolic support, but everything downstream is disconnected. So what happens is the axon downstream of the cut is going to start to degenerate. And these Schwann cells basically change from being myelin sheaths to kind of cleaning up the mess and making a guide for the axon to regrow and hopefully find its original place where it connected. So there's this word they call Wallerian degeneration. So the Schwann cells are also playing a role in helping this axon regrow and find its original connecting point after there's been damage and the axon got cut. You know, this is not something that works so well. In the CNS, if you cut axons, this is like where if you get spinal cord damage, you are basically quadriplegic or paraplegic because the axons don't regrow and find the place where they used to connect. But in the PNS, there's a lot more of a possibility for these things to regrow, especially if it's kind of a clean cut. If it's kind of a hamburger cut, then it's going to be trickier for the axon to find its little guide and reconnect to its original place. Another thing that the Schwann cells do, you know, I talked about how there are axons that are not myelinated if we don't care about the speed. But we still do care about short circuiting, right? If you have two wires that are touching each other, you're going to short circuit. So Schwann cells can play the role of insulators. Like I can have an axon and another axon, and they are being held apart from each other because the Schwann cell, it's not making a myelin sheath, but it is actually keeping them from short circuiting and kind of holding them apart. So typically you think of myelin sheaths as the purview of Schwann cells in the PNS, but they also can just hold unmyelinated axons apart so they don't short circuit. All right, so that is myelin in the PNS and the CNS. In the PNS, there are also another thing called satellite cells. I'm going to put this in for completeness. But we don't even really know what they do. They probably help, you know, kind of maintain the surrounding environment and cushion the neurons and stuff like that. You know, they get their name because, in fact, I should say neurons are large, glia are smaller and more numerous. So like in a ganglion where you have the neuron cell bodies, the satellite cells are all over the place. You can see they're kind of called satellite cells because they're like the little moons of, you know, orbiting around Jupiter. So this, you know, this would be a ganglion, right? Because collection of neuron cell bodies in the peripheral nervous system is a ganglion. So in the CNS, you don't have satellite cells. We have other types of glial cells that I'm going to talk about right now. But they still are small. So like in your brain, about half the volume of your brain is neurons because neurons are big, but you have like 10 times as many glial cells as neurons. So let's look at the glial cells in the CNS in more detail. like I said, oligodendrocytes making myelin. Another super important cell in the CNS tissue are the astrocytes. Astro, star, like astronomy. They look like little, or asterisks, they look like little asterisks. They do a lot of important stuff. They provide metabolic support to the neurons. Transfer sugars to them and stuff like that. They maintain environmental milieu. Or I should say maybe have an extracellular if you think about a few things. One, we know concentrations of ions directly affects membrane voltages. So we better make sure that the ion concentrations out there are maintained at the appropriate levels. We also saw that one of the ways that you clear a synapse is diffuse out of the synapse, right? Where do you go then, right? If I've got like, oh, here's some synapse and some neurotransmitter just diffused out, it's going to cause mischief if it gets into some other synapse and binds a receptor someplace else. So these astrocytes are basically vacuuming up stuff and clearing things out and making sure they're kind of like the custodians, making sure that everything is all kept at appropriate levels and cleaned up and all that. So astrocytes are doing that. Astrocytes help create the blood-brain barrier. So we've got our blood vessels coming in. We have, neurons and stuff out here. The little astrocytes grab on here. And they make a very kind of strict boundary between the bloodstream and the space where the neurons of your brain are. The reason why this is important, remember, when we were talking about caffeine, I said, oh, caffeine acts as an antagonist at adenosine receptors. So like adenosine, if it's in your brain, it's a neurotransmitter. It's going to bind receptors and it's going to mess with your, sleep-wake cycles and stuff. But in the rest of your body, adenosine is just like a building block, right? It's used to make nucleic acids and ATP and cyclic AMP or whatever, right? So the same molecule that is just a random molecule in your body, if it gets into your central nervous system, is actually going to be causing issues. So there is this really tight control. So tightly controlled, let me kind of write that more. Maybe, in fact, I'll type it out using a type tool to make it even prettier. Let's do it black. It'll come down. Very tightly regulated. Boundary between bloodstream tissue. So, right, most of the time your capillaries are really leaky. The whole point of a capillary is to deliver stuff to the tissues or to pick up waste to get rid of. So in general, capillaries are designed to be very permeable because the whole point is that's where you exchange stuff between the tissues and the bloodstream. In the brain and spinal cord, we have a different story. We have a really highly regulated boundary there and very special membrane proteins that only allow really specific things to move in or out of the bloodstream. Because if you just have random stuff getting into the neural tissue, it's likely to cause problems. This is an issue when you're designing therapeutic drugs that are supposed to work on the central nervous system. You might have some drug that is awesome. It's going to help somebody if it actually gets to the nerves or the neurons, but it doesn't get there because even if you inject it and it's in their bloodstream, it still doesn't leave the bloodstream and get into the spaces around the neurons. So this is a real challenge when you're designing CNS therapeutics. You have to come up with something not only that is going to be helpful if it gets to the neurons, but is actually able to be moved out of the bloodstream into the tissue there. So designing CNS therapeutics is way trickier than designing a normal drug because of that problem. Like one of the classic examples was like, like we'll talk about Parkinson's disease, where you have these neurons dying off that are releasing dopamine. And you can at least initially get some relief by increasing levels of dopamine in the brain. But dopamine doesn't cross the blood-brain barrier. But they found like if you give people this precursor, L-DOPA, that did get into the space around the cells. And then once it was there, it got converted into dopamine and helped alleviate some symptoms. But they had to kind of get a little roundabout. They couldn't just directly give somebody dopamine and have it, actually get into this, into the brain. So astrocytes playing important roles. And I should mention, astrocytes do more stuff. They have their own electrical signaling. It's slower. It's not like the fast spikes of the action potentials. Like an action potential maximum, they're spiking around like kilohertz, about 1,000 spikes a second. Astrocytes have electrical signaling, but it's much slower. But they are actually important they're doing more than just purely support. It's pretty clear they're like adjusting kind of bias points for, circuits in the brain by allowing more or less neurotransmitter to be like lying around in the spaces. And, I would say neurons get so much of the attention because they're the flashy, sexy things that are doing all the fancy like circuitry and spiking and all that. And a lot of the other things in the nervous system don't get as much attention. But that doesn't mean they're not like doing critical, it's like if you were gonna like read some like bestseller, it would probably be about the rock star, not about the secretary who made sure that everything worked out and that this person's career would have just, flatlined if the person hadn't actually been taking care of everything. But nobody's going to read, buy the book probably about the secretary or whatever. Like people's labs tend to get more funding if they're focused on kind of the fancier, sexier things going on. Let's continue. More glia. So So CNS glial cells, oligodendrocytes, astrocytes, the next one we're going to talk about are microglia. These are phagocytes. So they play a role in like immune response, eating stuff that shouldn't be there. They seem to play a role in actually pruning things and maintaining things. The central nervous system is what we call immunologically privileged. Your normal immune responses don't happen the same in your CNS. Some of that makes sense. Like if you think about a typical immune response is inflammation. There's some injury and there's inflammation. The last thing you need is your brain to get all inflamed because there's no place to swell, right? It's all located in this hard case in your skull. So a lot of the immune system stuff is specialized. So microglia are phagocytes. They're part of kind of the defenses, as well as it seems kind of cleanup crew and pruning and stuff in the CNS. Ependymal cells. If you've taken anatomy, then you know that the brain has hollow spaces in it filled with fluid, right? The ventricles and the aqueducts and things like that. We're going to be talking about those in way more detail. But all of the hollow spaces in the CNS, that your spinal cord has a tunnel going right down the middle of it filled with fluid, so the cerebrospinal fluid. So these line hollow spaces in the CNS. We're going to meet them. They're going to be also working with the capillaries to create the choroid plexuses that are actually generating the cerebrospinal fluid in the brain. They're kind of like columnar epithelium with little cilia. So they can help circulate the CSF in those hollow spaces. But again, on Thursday, actually, obviously not on Tuesday, we're going to be doing an exam, but on Thursday, we're going to be talking about the brain in more detail and talking about the ventricles and all that. And we'll meet these ependymal cells as lining the ventricles and the canals. We'll meet them as helping make the CSF and all that. What else should I say? I think that's it for now. I think we'll take a break, and when we come back, we have an enzyme lab to look at here. So, and I'll talk a little more about the exam as well. So let's come back at 10 a.m. and we'll continue.
Professor Eggert,
Uh-huh.
I don't think I made it to roll call, but I am here today, if you mark me absent.
No, I saw you come in at the end there.
Okay, thank you.
Okay.
a.m.
All righty. So let's spend a few moments talking about some of the main neurotransmitters and kind of classes of receptors, and then we'll move on to our lab. And so there's a whole bunch of different kinds of neurotransmitters. And there's 40 or 50 known ones, and people find new ones. And I am going to just kind of talk about some of the basic broad classes and some of the vocabulary you need to know as we go on. And some of these we have talked about. So acetylcholine is one of the common neurotransmitters. We usually call it ACH. There are two broad classes. So it's an adjective for things that are associated with acetylcholine is cholinergic. You know, if we're talking about a receptor that binds acetylcholine, it'll be a cholinergic receptor. If we're talking about a drug that mimics acetylcholine, it'll be a cholinergic agonist, or if it's blocking acetylcholine receptors, a cholinergic antagonist. So cholinergic just means having to do with acetylcholine. There are two main classes of receptors for acetylcholine. One of them that I've actually mentioned, the nicotinic cholinergic receptors. And the other kind are called muscarinic. They get their name cholinergic because they are cholinergic receptors. They bind acetylcholine, but then they have this differential part, nicotinic cholinergic receptors. Like in the lab, you can identify them because they bind nicotine. You know, these are what are binding. If you're smoking a cigarette, getting a little awake and a little more focused, it's because your nicotinic cholinergic receptors are binding the nicotine and turning on these attentional circuits. Muscarinic bind, it's called muscarin. It's found in those beautiful mushrooms that you find out in the woods that look, you know, the fly agarics, Amanita muscaria with the, you know, red cap with the white stars. Big difference between them, aside from this, is the nicotinic cholinergic receptors are directly gated. Sodium channels. So when we see something that has nicotinic cholinergic receptors on it, we know that there's only one thing that's going to happen. If the receptor binds acetylcholine, we're going to have the cell depolarizing as sodium comes in, right? So these are one-trick ponies. We're going to find these on skeletal muscle. We'll find them in a variety of other places as well in the nervous system. Muscarinic, these are indirect. So these are second messenger. What does that mean about the result of binding a muscarinic cholinergic receptor? What can you tell me?
There can be various types of reactions.
Exactly. Exactly. The thing you can tell me is you can't really tell me. When we get into the autonomic motor, both the somatic motor and autonomic motor have the muscles with cholinergic receptors, but skeletal muscle has nicotinic cholinergic receptors. So you know if you excite it, that the muscle's going to get excited and contract. For the smooth muscle with the muscarinic cholinergic receptors, you don't know what the result, whether it's going to actually contract or relax or whatever. It depends on the situation you're looking at. So this is important. Acetylcholine, cholinergic. Let's look at some more kinds of neurotransmitters. So biogenic amines, that means things that are a few steps from amino acids. So like if you want to make serotonin, you start from tryptophan. And there's the indolamines, like serotonin. If I can. This is from tryptophan. There's the catecholamines. Catecholamines are derived from tyrosine. It's just another amino acid. This includes things like dopamine and epinephrine, and norepinephrine. So catecholamines, these are the ones, and these are the ones that are more kind of energetic, so to speak, right? Epinephrine, another word for epinephrine is adrenaline. Another word for norepinephrine is noradrenaline. Dopamine is the one that's found in a lot of these kind of reward circuits. It's also, it gets implicated in a lot of addiction and stuff as well, kind of where you have, kind of these reward circuits getting kind of derailed towards behaviors that aren't as helpful. Let's talk about these two particularly, epinephrine and norepinephrine. Epinephrine is also called epi. Norepinephrine is also called NE. And again, epi is adrenaline. It's just synonyms. If we were in London right now at some college, I would say, and the catecholamines include like adrenaline and noradrenaline, or however they talk. So adrenaline and epinephrine are just synonyms. In general, noradrenaline tends to be more used as a neurotransmitter. So in synapses, whereas epinephrine is more of a hormone dumped into the bloodstream and delivered that way. However, they often bind to the same receptors. Things that are associated with these guys are called, so the adjective here is adrenergic. And again, that's why it's important to know that epinephrine is also called adrenaline, because otherwise adrenergic doesn't make any sense. So adrenergic, meaning pertaining to epinephrine and norepinephrine. And there are adrenergic receptors. There are alpha adrenergic receptors. There are beta adrenergic receptors. There's alpha 1, alpha 2, beta 1, beta 2. These are found localized in different places. Some of them play slightly different roles. Actually, one of them is actually a presynaptic receptor. I'll talk about that more later. But like when you think about beta blockers are prescribed to keep somebody's heart rate down if they have high blood pressure. A beta blocker is just an adrenergic antagonist that is specifically working at the beta adrenergic receptor. That's why it gets its name beta blocker. It's blocking a beta adrenergic receptor that's on the heart that speeds up the heart and increases contractile strength and stuff like that. So adrenergic, pertaining to adrenaline or noradrenaline or epinephrine and norepinephrine. Epi, like an EpiPen, if you jam someone with an EpiPen, you're basically jamming adrenaline into their thigh. All right, so we'll come back to this as well. Other kinds of neurotransmitters. So this is neurotransmitters continued. Some are just amino acids. And not necessarily, right, there's lots of amino acids that aren't the 20 amino acids that make up all the proteins in your body. So these are amino acids that are important neurotransmitters. One of the most important excitatory neurotransmitter usually involved in excitatory pathways is glutamate. Another important amino acid that acts as a neurotransmitter is called GABA. Gamma amino butyric acid. This is inhibitory. This opens chloride channels. Right? If you're opening a chloride channel, you're going to bring the voltage down and you are going to take the neuron, the target neuron, further away from its threshold. In terms of the receptors, so some of the glutamatergic receptors, NMDA receptors. Other ones are called AMPA receptors. Don't worry about what NMDA is, N-methyl D-aspartate. Again, it's kind of like muscarinic or cholinergic. It's just a way to distinguish between different kinds by looking at things that they can bind. The main thing is to know if you're seeing an NMDA receptor, that it's a glutamate receptor. For instance, like one of a common, an anesthetic, actually in club drug, and now actually used as an antidepressant, ketamine. Ketamine is an antagonist to NMDA receptors. So if somebody is got ketamine in their system, it's blocking these glutamate receptors, and it means these messages aren't moving from one place to another. That's like, the K-hole, people feel completely dissociated from their body. They're like a consciousness and don't even feel a connection to the rest of their kind of corporeal form, because those messages from your body aren't making it into other parts, deeper parts of your brain. So NMDA, NMDA receptor is glutamate. GABA, one of the interesting things about the GABA receptor So this is bringing up, so most of the time when we describe stuff in this class, it's always a simplified version of reality, which is always way, 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 way more complicated. So a lot of these receptors that we're talking about, they sometimes have other binding sites on the receptor that isn't for the primary ligand. So like a GABA, the receptors that bind GABA, they have a place to actually bind the main ligand, the GABA molecule, but they have other binding sites that actually modulate the activity of the receptor when you actually bind GABA. So this site that I put here, this is where the benzodiazepines bind. And so if a benzodiazepine, if like Xanax or Valium or one of gets in there, what happens is when GABA binds, it'll stay open longer and will let more chlorine through and create more of a negative swing of the voltage. Like the benzodiazepines aren't actually directly agonists or antagonists. Instead, what they're doing is they're binding another site on the receptor that makes the channel stay open longer. So when we have this moment where GABA is binding, not only does it open chloride channels, but it lets more chloride in than it would have if the benzo hadn't been bound onto that other site on the receptor. So just kind of giving you a sense, it gets a little more complicated. You can have these kind of modulatory binding sites that are not the main ligand binding site. So more. Heck. There's a lot of peptides that act as neurotransmitters. Again, peptides are just like a few amino acids connected together. A lot of the things in the pain pathways are peptides, like the naturally occurring like endogenous opioids, and a lot of actually the things that the hypothalamus uses to control the pituitary and stuff. There's a lot of just very small things like 7, 8, 10 peptides that are really important signaling molecules. So peptides are important. There's lots of others. There's gases. One of the important neurotransmitters people found out was nitric oxide. Even carbon monoxide, which is a deadly poison if you breathe it, in small amounts in the appropriate situations can be a signaling molecule. What's interesting about these are going to be, obviously, you're not going to keep a gas. This is nitric oxide. This is carbon monoxide. Those aren't going to be stored in vesicles. Instead, you have little enzymes that are turned on and make the gas. And then in that moment, the gas can diffuse and bind to receptors someplace else, but it's not released by vesicles fusing through exocytosis. It's actually just generated by some enzymatic reaction like the oxygen bubbles in our lab. Then it can diffuse across the membrane. And in fact, this one often acts as a retrograde neurotransmitter. You know, the main thing is releasing neurotransmitters that bind receptors here, and then this thing is sending messages back saying handshaking, yeah, I got a message. Right, so this idea of synapses being one way is wrong. And also there are often autoreceptors, like some of those, like in some of those adrenergic receptors we're talking about are actually, not only are there adrenergic receptors on the target, there's adrenergic receptors on the very neuron that's releasing. And you can then have feedback saying, okay, we're releasing the neurotransmitter, but we've got enough because I can get the message here that there's a whole bunch in the synapse already, so I can stop releasing it. Right, so this idea of where you find the receptors for the neurotransmitters, the neurotransmitter receptors might actually be on the presynaptic terminal. So just kind of putting that out there as well. All right. I'll get anandamide. I'll give you. This is kind of a fun one because when I first started teaching this class, this didn't have a name. It didn't exist. When people were trying to figure out why do people get high when they smoke cannabis, they knew THC, tetrahydrocannabinol, was the kind of active ingredient, but they didn't know why it made people high. And they thought, well, it's lipophilic. Maybe it's getting in the membrane and messing with the opening of channels. And because there are certain anesthetics that actually do seem to work that way. They get in the membrane and then affect the dynamics of the other membrane proteins. But what they eventually found was, nope, THC is actually binding receptors on neurons, and actually not just on neurons, but in cells all over your body. You know, and they called them cannabinoid receptors because they were binding, you know, this stuff from cannabis. And then they figured, well, if your brain is filled with receptors that bind THC, there's got to probably be an endogenous ligand that they are binding when you're not getting stoned. So they looked for it, and sure enough, they found it. And then this is, you know, not all scientists are, well, some scientists can be fun. Ananda is Sanskrit for bliss. So they named the endogenous ligand that binds the cannabinoid receptors after bliss. But it's just another neurotransmitter now that people are still trying to figure out all the ways it plays into. Obviously, it plays roles in memory. Think about people get stoned, start like, what am I doing? Or pain, it's definitely plays roles in modulating pain pathways. And it seems to play a lot of somatic roles as well, not just kind of nervous system. It's like on all sorts of cells, not just neurons. But just kind of putting that out there. You know, this didn't even exist until somebody thought to look for it. So what else? Just kind of putting that out there. We're going to be looking at different neurotransmitters during the semester. In particular, we'll be seeing acetylcholine and epinephrine and norepinephrine, in particular when we're looking at the sympathetic and parasympathetic, I should say autonomic as well as somatic motor. The other ones we probably won't look at in as much detail, but it's kind of nice to have them all in having context. All right, any comments or questions about any of that? Just to kind of give you a little bit of a lay of the land of, and I should mention that just because something is a neurotransmitter doesn't mean it doesn't have other roles, right? Serotonin, which you now usually think of as, oh, that's like this neurotransmitter in your brain and related to depression or this or that. When people first discovered it, it was important in looking at how, like, you stop bleeding when you're cut, hemostasis. We're going to meet serotonin when we look at platelets a little later. This idea that serotonin was an important neurotransmitter in your brain, that just seemed crazy. Serotonin is also an important signaling molecule in coordinating your kind of digestive processes and all these little nerve plexuses. Serotonin plays different roles. Okay.